Hello, I'm Jonathan Fletcher from Brigham and Women's Hospital in Boston, and I'm honored to coordinate this new Liddy Shriver International Research Consortium for Leiomyosarcoma. The lead investigators for the consortium are from France, Germany, and the United States. This program is a coalescing of expertise with the goal of moving as rapidly as possible from new research lab discoveries into new clinical trials. As outlined here, the consortium combines complementary expertise, which is needed to identify new treatment targets for leiomyosarcoma, and then concurrently find the drugs that will make things happen in people. Importantly, the investigators have expertise in so-called molecular subtyping of leiomyosarcoma. These are tools needed to identify which patients will benefit the most from new therapies. The group also has expertise in functional genomics. These are exciting new tools in which each of the 20,000 genes in the human genome can be knocked out one by one in leiomyosarcoma cells. These studies will tell us which of the genes are the most indispensable to leiomyosarcoma. Drugs targeting those genes can then be evaluated using a number of leiomyosarcoma laboratory models that have been developed uniquely by the investigators in this program. As you can read in this program overview, we are focusing particularly on a biological pathway which is called PI3K. This is a pathway which is known to be crucial to many leiomyosarcoma cells. Unfortunately, a number of pharmaceutical companies have developed drugs which shut this pathway down. Unfortunately, clinical trials with these drugs, including leiomyosarcoma patients, have not shown dramatic activity, clinical activity for most patients. Therefore, as an urgent priority, we need to do the biological studies which will tell us how to use these drugs more effectively. I hope you have time to watch the videos from the other lead investigators in this program, including Drs. Van der Rijn, Schibone, Marinho Enriquez, Bauer, and Langenau, because they highlight other exciting opportunities that have emerged already from the program. Most importantly, the investigators would like to thank 3 sarcoma foundations without whom none of this would be possible because they have provided the research funding. They are the Liddy Shriver Sarcoma Initiative, the Leiomyosarcoma Direct Research Foundation, and the National Leiomyosarcoma Foundation. The investigators would also like to thank our many clinical colleagues who have advised us in the development of this program. We'd particularly like to highlight Drs. George Dimitri and Suzanne George from the Dana-Farber Cancer Institute and Dr. Jean-Yves Lay from Lyon in France. These and other colleagues have agreed to advise us as the program develops, meeting with us yearly to ensure that the best leads from the research can move quickly into clinical trials for leiomyosarcoma patients.